Hey there, ladies and gentlemen, boys and girls. Today we're going to talk about something called nondisjunction. All right, and basically this is what leads to things called chromosomal disorders. Basically, this is what happens when things go wrong. So this is what we're going to do today. We're going to do this kind of a couple different phases. I'm first going to talk about healthy meiosis, so that we can compare it to what happens, compared to what's supposed to happen versus what might accidentally happen. Just like we did with DNA, we talk about how DNA is supposed to be read to make into a protein, and then we talked about mutations, and that's where there's a problem in that process. All right, and so we're going to talk about those. First is autosomes, then we'll briefly talk about it in terms of sex chromosomes, right? And then we will talk about chromosomal notation when we look at a karyotype. So there's kind of three phases of this today. So, and I'm obviously going to be kind of going over very quickly a lot of the details because we're going to highlight what happens when it goes right versus, more importantly, what happens when it goes wrong. That's our focus today. All right. So let's focus about what happens when it goes right. So I'll give you guys a moment if you have your notebooks out to kind of copy this down. I highly recommend pausing the video, kind of set yourself up. Okay, so I'm hoping you did that now. So let's talk a little bit about what happens when it goes right. So what we have here is kind of early on, we're starting with our one diploid cell. All right, we're starting our third diploid cell. It's got two sets. We got the paternal set and the maternal set. Notice we have pairs, right? I'm only doing 3 pairs. We know that we have 46 chromosomes, right? 46 chromosomes to start, right? And we've gone through DNA replication. So for that number 1 chromosome I got from my father, or from the maternal chromosome, I have 2 copies of it. But we count it because we count by the number of centromeres, the center part that holds those together. That's 1 chromosome. All right, I have 2 copies, right? So I have 23 pairs, 2 sets, right? And even though I have 2 chromosomes, I have 46 chromosomes, I have 92 chromatids because we've gone through DNA replication. All right. So at this point right here, early on, we have our things like what we talked about going through our tetrad formation. They've lined up as tetrads, right? We have our crossing over, tetrads where the homologous pairs line up next to each other. We have that crossing over, which is that switching of those similar genes, right? And then they're eventually pulled apart. And that first stage there, meiosis 1, is about separating those sister chromatids. Not sister chromatids. It's about separating those homologous pairs, which we would also see the crossing over at that point. So we have the number 1, the number 2, and the number 3 over here. In this cell, number 1, number 2, number 3 there. And basically what we've done here is separate the homologous pairs. That's what we're supposed to do there. All right? And so now we have, since each one has just one number 1, one number 2, one number 3, we now have 2 haploids. Right? And then we go into meiosis 2. In meiosis 2, what we're going to do, kind of a similar process where you're going to now separate the chromatids. All right, we separate the sister chromatids. So we pull apart each one of these sister chromatids, the number 1, the number 2, the number 3. And so what we get here is kind of one chromatid here, here, here for the number 1, 2, 3, and then we have the other side here. Here's the DNA for number 1, number 2, number 1, number 2, number 3. We would do the same thing with this one over here. We have the number 1, the number 2, number 3, and we pull apart these sister chromatids to get the number 1, number 2, and number 3. And what we end up with at the very end is 4 haploid cells. Now haploid because they each have one number 1, one number 2. They don't have 2 up here like we do here. We have 4 haploid cells, or 23 chromosomes in each one. That is what is supposed to happen in meiosis. This is healthy meiosis happening here, right? Non-disjunction, which is what our focus is today. Non-disjunction is what happens when this process, there's a malfunction. And it typically has to do with one of these parts too right here, all right? It either happens with the separation of the homologous pairs or the separation of the sister chromatids, all right? And so what you might see there is a change because of that. So let's kind of go through it. Let's set an example in terms of the sister chromatids, or a separation of the sister, separate the homologous pairs. So we know that at this point right here, we're supposed to separate these two, all right? So we should get something like that, all right? Something like that. And then what we have is we have an error here. All right, and it normally happens to do with a mutation in the centromere that holds together those sister chromatids. That paper clip that holds those those strands of DNA together, it doesn't break properly. And so what happens is when we're supposed to split the sister chromatids, right? What ends up happening is maybe, the number 1 splits okay, the number 2 splits okay, but the number 3 doesn't. Notice that those are still together, which means these didn't pull apart. They both went to one side, which means on this side here, I'd have the number 1, right? The number 2, but no number 3 because both strands went over here, right? And then I could say the same thing for the other one. What happens if, we separate these ones and get 1, 2, 3, right? Oop, no, let's say we don't get that one there. Let's say the number 1 right? And what happens is here, now I all of a sudden have 2 copies of the number 2 chromosome here and none here. Now remember, the whole purpose of this is to create or combine 2 gametes, right? To create 2 sets. So the idea being, it wouldn't be the same ones, but you get 1 number 1 chromosome from your mom and 1 from your dad. But if I were to use 1 number 3 chromosome from my dad, 1 number 3 chromosome from my mom, but if I use this one right here, if I use this gamete, I will get 1 number 1, 1 number 2, and 2 number 3s. Which means if this is a sperm cell, I get 2 number 3s from dad, and I would get 1 number 1 from mom. That's 3 chromosomes. I'm supposed to have 2 for each one, not 3, which means that would total up not to 46 chromosomes, but to 47. And our cells work on a very specific amount of chromosomes. It can lead to a lot of problems, and a lot of those problems we call genetic disorders. Some of them are a little bit more common that we're familiar with. Things like Down syndrome is caused by an excess chromosome. We're going to talk about that in a moment. All right. This is what it would look like if it was in terms of a autosome here. All right. So what I'm going to do real quick is I'm going to pause it, reset it. We're going to talk about real quick about sex chromosomes. That was bad timing with the bell. All right, so I've reset the board a little bit here, but now I just want to focus on that 23rd pair of chromosomes, the sex chromosomes, because those ones will define gender. All right, so I'm going to use me as an example, being a male. Genetically, I am a male, and so genetically, I have an X chromosome and a Y chromosome. That Y chromosome determining me to be genetically male. All right, now we know I have 1 X, 1 Y. But we know that when a cell goes through cell division, that it has to make a copy of all of its DNA. So for a brief moment, I have 2 copies of that X chromosome, right, and 2 Y chromosomes. Briefly, I don't have 4 sex chromosomes. I only have 2. But remember, we go through DNA replication. So I have 2 X chromosomes and 2 Y chromosomes. I'm going to, instead of drawing the little squiggly lines like that, I'm going to draw the letters just to make it more clear when we're talking about the sex chromosomes. All right, which means I'm going to separate the homologous pairs. So the X's would go here, those Y's would go here, and then I separate the sister chromatids. So I have an X here, Y here, a Y here. Now as a male, all right, these would end up becoming sperm cells, and then it would be a combination of the sperm cell and the egg cell. I would give up one of my sex chromosomes. If I give up one of the sperm cells that is a Y chromosome in it, I will have a son. If it's one that has an X chromosome in it, I will have a daughter. All right. Nondisjunction is a problem with the separation of these chromosomes. Right. So I give up a Y. My wife would give up an X, and thus we would have an X and an X, or X and a Y, and have a son. All right. Nondisjunction, on the other hand, is the problem with the separation of that. So we go through DNA replication. We have two copies. All right, they split here. Right? But now it's a problem. We have an error. We have a problem with the separation. So maybe we're supposed to have one X in here. Maybe we have two copies, which means there's none in here. And then maybe we have the separation of the Y is okay. All right? Which means if my wife gives up an X and I use this gamete, she gives up an X and I give up two X's, that's 3 X chromosomes. You're only supposed to have 2. All right, that leads to a lot of disabilities. Sometimes, depending on where the nondisjunction takes place on your chromosomes, it can lead to just straight up death of that organism. They don't go past that zygote phase. It wouldn't even be death because they never really form properly. All right, and so I want to take a moment now, kind of stop there, and we're going to talk about what this looks like when we get it into a karyotype. Okay, so what I have here is a karyotype, all right, a laying out, an arrangement of our chromosomes based on size, right? We have our homologous pairs here, all right? I don't expect you to copy down a picture of a chromosome, but we're going to talk about some of the notation that we see here. This is a healthy individual in terms of the number of chromosomes. So we have 2, 4, 6, blah, 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 up to 46, right? 2 number 8s, 2 number 9s, 2 number 10s, and then we have one or two sex chromosomes, right? This means meiosis went well, and both fertilization went well. All right, so this is 46 chromosomes. I want to show you guys a little bit of notation. The way that I would write this notation, all right, because then we'll look at what it looks like when it's not. This is what we call chromosome notation. All right, there are here 46 chromosomes. So there's the number of chromosomes. All right, and then you would write, all right, what the sex chromosomes are. In this case here, I have 46 chromosomes, XY, and then there's another one here when we have mistake, when we have nondisjunction, all right, which we'll talk about in a moment. But that's the normal notation. This is kind of the healthy. So we have the number of chromosomes. We have the sex chromosomes. In this case, this person has 46 chromosomes, and they have an XY, so they are male. All right. So now let's look about what it is when we have a mistake. All right. So here we are. All right. There was a mistake in the mutation here, and one of them has too many chromosomes. We have 2 number 1s, 2 number 2s, until we get down to this one right here. All right. There are 21 is a small chromosome, and there are 3 of them. All right. There are 3 of them. All right, which means my notation has changed. All right. This is kind of the healthy notation, but now we have a new notation here. All right. For this individual here, the way that this would work is I have here not 46 chromosomes because I have 2 of each except for an extra one here. So now I have 47 chromosomes. This individual is an X and a Y, so they are male. All right. But now we have to add a third one here because there has been a mistake, and it highlights or identifies this mistake. We have an extra 21. All right. We have an extra 21 here, which means we would write plus 21. And that tells me which one we have that is missing, or that we have added extra. All right. So now we have a 21, and that 21 tells us that it's on the autosome. In this case here, this one, because there are 3 of the number 21, this is also known as trisomy 21. Oh, sorry. Trisomy 21. Trisomy meaning 3. All right. 3, number 21. So this is a trisomy 21 here. All right. So let's look at another example. Okay, so here's another example here. This is actually one that's gone. This is a good picture I had from. This one's already gone through DNA replication, so we have the chromatids. All right, but notice we have 2 of them, all of them, except for right here. This is another autosomal one. I have only one number 20. So in this case here, I do not have 46 chromosomes here. Let's go with my notation. I have 45 chromosomes. Again, I don't know why I'm picking more males. It just happens to be, that's just a coincidence. All right, XY. But this time it's not plus 21 because we don't have an extra one. This time we have a minus 20. All right, so that whatever number goes right here tells us which one we are missing or have too much of. This one is also not a trisomy because we only have one. This one would be called a monosomy. Oops. Monosomy 20. Because a monosomy meaning 1 of the number 20 chromosome. All right, let me show you guys one more example here using the sex chromosomes. All right, so this one is a little bit different because the other 2 I did were all based on the autosomes, number 1 through 22. This one is someone that is missing a sex chromosome. They are missing one of them. And so this one's a little bit different, all right? This is sometimes, in this case here, I only have one. This is called a monosomy X. The notation's a little bit different here because this one is on the sex chromosome. All right, in this case here, it is we have 45 chromosomes. All right, not 46. All right, and in this case here, the second one is always based on the gender. This one is technically female because there is no Y, but we would just write as 45X. This is actually Turner syndrome. All right. Now there's this next one, last one here would go with if it was a autosome, but there's nothing that's going to go here. This is just sometimes called a monosomy X. Because there's only one X chromosome. Right? You can never have just a monosomy Y. There is no such thing as that. The Y chromosome doesn't have enough genes on it to allow that organism to live on. Okay? All right, I know that was kind of a lot there, but I wanted to show a couple examples as well. Nondisjunction is what happens when meiosis goes wrong, which leads to an improper number of chromosomes, and then we just have different notations to show that. All right? Otherwise, thanks for watching, guys. I know this was a little bit of a long one. Good luck, and may the science be with you.